we've looked at DNA replication in some detail. We're going to take a step back now and actually look at cell division. Right? And this process we usually referred to as mitosis. Now, mitosis is really only part of what's called the overall cell cycle. Right? So the cell spends its time divided between different phases. Okay, so we have an M phase, which is the mitotic phase. So M is mitotic. That's where meiosis is going to occur. All right. And then what we're going to have are a series of growth phases. So we have a G1 phase. So there's the M phase. Then there's the G1 phase. And so G1, simply growth. That's what the G stands for. And what's going to happen during this time is a lot of protein synthesis. There's going to be the doubling up of a lot of material within the cell, all right, because essentially the cell is going to be in a process of dividing and dividing and dividing over time, and it has to be getting ready to do that. Okay, so this is just post-mitosis. The cell had just divided, and now it needs to produce more proteins. Eventually, it's going to get to the point where it's going to start to be ready to divide again, and we enter into what's called the S phase. So the S phase is the synthesis phase, and that's really DNA replication. So that's what we just finished kind of going over. DNA replication is the synthesis phase. And so that you go back if you haven't looked at that yet. And then after the synthesis phase of doubling up the DNA, we still have another growth phase where additional proteins are doubled up in preparation for the actual division process. And then we enter the M phase, the mitotic phase. So what we're going to do now in this particular lecture is really just look at the different phases of mitosis, just so we're going to be primarily focused here on, for us, just the M phase. Right? We already covered the S phase. That, like I said, was synthesis or DNA replication, and now we're into this phase here. And then later, after that, we'll start into gene expression, transcription, translation, which is a lot of protein synthesis, which is really what's going on in the other phases, in the G phases. So what do we have here? Here's a series of little sketches of cells with chromosomes at different stages of mitosis. And at this point, what you should be able to do is, or at least not quite at this point, but after this point, is be able to sketch this yourself just from scratch, for one, and that would be the best way to do this. Just get a blank piece of paper, draw cells, draw nucleus or chromosomes, and then label them with the particular stages. If you can do that, then if you're just shown a picture of it or you're looking at it under a microscope, it's going to be a lot easier for you to actually identify the stages, and that'll be one part of what you need to know. So the first thing is a stage called interphase. Now, if we're looking at this cell cycle, right, the cell spends really most of its time in interphase. Okay, so out of this whole cell cycle, all this, would really be interphase. Right? So all this time, okay, except when it's actually getting into the true mitotic stage. So this is the way the cell would look most of the time. And so in this drawing, what we're representing here is the outer cell membrane. And then inside here, this is supposed to be the nuclear membrane, and the nucleus, I just mostly filled it in. Essentially, if you were to look at this under a microscope, it would look sort of a solid color if it were stained. Okay, so typically when you're looking at a slide, when you see photographs of a cell, you're going to see the nucleus, and it's going to look like a sort of a solid color. It's not actually colored. That's because it's been dyed, and that's why it'll look different colors in different photos that you might see. But the chromosomes in this point are spread out. And so it makes the nucleus, when the dye is there, look almost a solid color. And that's what we're going to be kind of focused in on, is what it looks like at these different stages if you were to look at it under a microscope. Now, people just have made up these names and these stages, so there's kind of a little bit of, when does one exactly end and begin. Some of them have very distinct characteristics, and others kind of blur from one into the next stage because there's action going on, there's movement happening within the cell. But we have some very specific characteristics that you should pay attention to. So interphase, how does it look? The cell looks like it has pretty much a solid colored nucleus, at least when it's for a stained cell. If you're to continue looking at a group of cells, and then you were starting to see some that in the nucleus, you can see these little squiggly sections and some clear sections. Right? So these things here are representing the actual chromosomes. And they would become visible. So the chromosomes are visible. And that is what we call prophase. And so another characteristic of prophase to take note of is that you can still see the nucleus. Now, typically in these stains, the nuclear membrane itself isn't stained, but it's just the idea that there is some sort of invisible barrier between the chromosomes and the cytoplasm. And although you may not be able to see the barrier itself, you can tell when something is pushed up against the barrier. And so that's the idea. The chromosomes are inside the nucleus, and you can kind of see their organization is still within this circular framework. Right? They're still within the nucleus. So prophase, visible chromosomes, but contained within a nucleus. We then enter into the next phase, which different places you'll see it called different things. Sometimes it's completely skipped over, but I think it's really an important part, because later on, as we get into the next lecture where we talk about the actual cytoskeleton and the connection and what actually makes these chromosomes move and separate, this phase will have some unique and important things to it. So we call this Prometaphase. So kind of prophase, and then the next one we're going to get into is called metaphase, and this is sort of in between those two. There's a couple unique things that are visually distinct about this stage, and then there's some very important events that are going to happen during this event with the microtubules that are going to be attaching to the chromosomes and the sister chromatids. So what does prometaphase look like? How does it differ? Well, the thing is, this nuclear membrane is going to break down. Okay, so the nuclear membrane will break apart. So you can see how pieces here, of nuclear membrane. Technically, they do stay intact in chunks that are then controlled by other aspects of the cytoskeleton, so they can be reassembled later on. But they're broken up, and the chromosomes are set loose inside the cell, and they're not organized in any particular way. They're just free. So you can still see the chromosomes. They're visible chromosomes inside the cell. But the difference here is that they are not held inside a nucleus. They are disorganized and spread through the cell. So chromosomes are disorganized and spread out. Right, it's during this stage where these kinetochore tubules, which we'll get to, are going to attach actually to the centromere of the chromatids to begin to organize them into what we see coming next in the next phase. So without this phase, we don't really get the next phase. The next phase is called metaphase. Metaphase is one of the most distinct phases, sometimes fairly easy to identify, maybe a little more so than some of the others, and that we typically describe it as seeing that the chromosomes are lined up along the center of the cell. Now that's the thing that's misleading if you don't get into the next lecture, the material I'll cover next after this, is how does that happen? A lot of introductory courses just go over this basic process and don't go into the cytoskeleton, and then you're left thinking that somehow the chromosomes organize themselves, that they move to the center of the cell. That's not what's happening at all. But that's what it looks like. Right? The appearance of metaphase are chromosomes lined up in the center of the cell. This is really the result of an action taking place where at two opposite sides of the cell are things called microtubule organizing centers. Right? They're these little machine type areas that are building microtubules, and they're sending out these tubules that are attaching to the sister chromatids. And you essentially have these tubules attaching to the sister chromatids from two opposite MTOCs, or microtubule organizing centers. And what's going to happen is they're going to pull on them. So these things are in place. This is what happens, see, during prometaphase. And so during prometaphase, they're starting to get sent out like this, you see. And they're getting sent out then from the other side as well. And then what's going to happen is the tubules from each MTOC are going to be pulled or shortened. And there's sort of a tug of war that's going to take place. This microtubule organizing center is going to try to pull the chromosomes toward it. The other microtubule organizing center is going to pull the chromosomes in the opposite direction. Neither one is stronger than the other or having a greater pull. And the sister chromatids, so what we're really looking at here, so in this particular picture, you know, what we're really seeing is my little squiggle down here is representing something like this. So what you're looking at is this would be technically a chromosome, but in this case we call them collectively, these two of them, these are sister chromatids. Each of these, so each one of these is double-stranded DNA with all of its associated histone proteins. So we have DNA and histone proteins making up a chromosome, and that's what you see here. So this is actually the result of DNA replication. At the end of DNA replication, you have two new double-stranded DNAs. These, so here's one double-stranded DNA, here's a second double-stranded DNA. That's the result. Now the thing is that happens is that there's a protein that binds the two together, okay, at this region here called a centromere. So the two new double-stranded DNAs are held together and they cannot be pulled apart. And so I said we'll get more into the details of the proteins in the next lecture, but it's important to kind of put these two things together. So during prometaphase, when these tubules attach, they're attaching, from one microtubule organizing center here, from another microtubule organizing center here, and then they're trying to pull the sister chromatids away. The two new double-stranded DNAs are trying to pull them away, but they can't be separated because they're held together. And so that creates sort of this tie, between both sides, and it lines them up in the center of the cell. So you get metaphase. So metaphase in the middle, you see the chromosomes. Ultimately, a signal is sent that releases this protein. And when that protein then lets loose each of the sister chromatids, then the individual chromosomes can be pulled to opposite sides of the cell. So in this stage, this next stage, they're going to each of these MTOCs, this next stage is called Anaphase. And so during anaphase, it's a stage of motion or movement. Now, as you look at this, say, on a slide or a photograph, it just looks, it's a still picture. But anaphase is a phase of movement, so there's no one exact look of anaphase. Anaphase begins the moment the sister chromatids are pulled apart, so they can be very close together. And as they're being pulled further and further away from one another and closer to the opposite sides of the cell, it's still anaphase. It's all anaphase. So you could see them very close. You could see them almost at opposite sides of the cell, and it's anaphase. So that is a stage of movement, right, of the chromosomes apart. After anaphase is over, and anaphase is usually quick. Metaphase can last for a while, and some cells can arrest in metaphase, which means that they can get to this point and then stop and then just stay there. And they can stay there for a very long period of time before they move forward. Fertilization in some organisms actually takes place during metaphase, and then the ova continues to divide afterward. Right? So Metaphase is a stage where you can often see it because the cell stays in metaphase for a while. Most of the time, if you look at cells of a tissue, you're going to see them in interphase because, like I said here, interphase takes up most of the time for a cell's cycle. And then it enters the mitotic phase, and then you run through these. Metaphase of them is one of the longest. Anaphase is quick, so sometimes it's hard to find. Now what's going to happen is the chromosomes are going to be pulled to the opposite sides of the cell, now that they're separated, and we begin two things that's what are going to happen. Telophase is how we describe what's going on with the chromosomes. So the chromosomes in telophase are on opposite sides of the cell, and they have begun to what we call decondense. So we can't quite see individual chromosomes anymore. The proteins that have one called condensin that has tightened them up, all right, and made them actually visible. They're not visible on purpose, like for us to see them. They're visible because they are so compact, they're so dense that we can actually see them through a light microscope. Otherwise, they're more spread out and we would not be able to see them through a light microscope. But that's not the reason why they do it. They do it because it's easier and better to organize and not get twisted and tangled, and it's easier to separate them if they are more condensed. So in interphase, they're really spread out, which is why it looks like it's almost solid, because the chromosomes are sort of everywhere. Before division, they get really organized, and so you can actually see them as these little clusters, right? The actual chromosomes that we see visibly during prophase. But now, as we enter telophase, they start to loosen up again. What also starts to happen during telophase is the nuclear envelope begins to reform. So we now get reformation of the nucleus in each side here. And then we get the cells. Well, the cell itself is now going to start to divide into two new cells. And that is somewhat of a separate process controlled by different proteins. It's tied to the timing of the mitotic separation of chromosomes, but it's a process called cytokinesis. So cytokinesis is the actual separation of the cell. And what we've really studied, when we talk about mitosis, we kind of combine the two together. But the process that we have just looked at is technically called Karyokinesis. So if you think about the names of cells, we often refer to them as being prokaryotic or eukaryotic. Karyo means nucleus. And so karyokinesis is the division of the nucleus specifically. There are cells of a number of different organisms that at times undergo karyokinesis without cytokinesis. So what does that mean? That means they undergo nuclear division without cell division. So it's essentially one cell with one nucleus. The nucleus divides, and now it's one cell with two nuclei. And that happens, say, in embryonic development of insects. They actually go through multiple karyokinesis events without cytokinesis, so you have a multinucleate cell, one cell with many nuclei. And you can see this in fungi, and you can see this in a number of different organisms that happen. So they are kind of two separate processes, but they are coordinated together under mitosis. So it is, so cytokinesis occurs right after the mitosis. So we could actually put it in here. Cytokinesis, right at the very end of the mitotic phase, then the cell would divide, right? And there are other proteins, these actin filaments that kind of wrap around the cell along the midline of the cell, and then they kind of tighten, they squeeze kind of like a belt, and they pinch the membrane down, and then the two cells will actually separate into new cells. Then it's going to have to go into a growth phase again, because the cells aren't going to keep getting smaller and smaller and smaller. They have to stay the same size. So the cells have to build back up membranes. They have to build back up enzymes, all the proteins in the cell, the enzymes, everything has to build back up again. Then they can eventually get ready to go through synthesis again to make more DNA, go from single chromosomes to double sister chromatids, build a few more proteins to get prepared for the division process to occur, then they'll enter the division. And then this kind of happens over and over and over again as cells keep dividing. So you need to know the names of these phases: interphase, prophase, prometaphase, metaphase, anaphase, and telophase. Be able to sketch them out. You should be able to identify sketches of them if they're placed in front of you. You should be able to explain in general what's happening during each of these stages at this point with the chromosomes. The next step of this will be the next following up lecture, where we're going to talk very specifically about these microtubules, the names of the tubules, how they're formed, a little more detail. So we're going to kind of take this process and add an extra level of detail to it. And that's all for the introduction to mitosis.